So apologies to those of you who saw this last year. It will be quite similar, but if you did, then I would recommend you pull up the actual Jupyter notebook and see if you can work through it. So this is just to show this. I've been using Colab a lot for teaching in my class. It's nice because you can actually get access to a GPU, which is very useful for teaching these machine learning things. So I'm going to give a bit of context to the problem that I'm going to be developing a model for using this Pyro probabilistic programming language thing. So to set the stage for that, my lab works on, like many labs in genetics, trying to understand the links between genetic variation and human genetic disease risk, and particularly trying to figure out potential molecular mediators, one of which is RNA splicing. So as you probably know, human genes contain exons and introns. So introns get removed from the initially transcribed messenger RNA to give these different splice isoforms. So variation in what gets removed as intronic gives you different isoforms, can give you different proteins, and actually can also regulate gene dosage. And there's an increasing understanding that this can be one of the ways by which genetic variation impacts disease risk. So I got into this space developing this algorithm called leafcutter during my postdoc, which quantifies splicing variation from short-read RNA-seq. I'm not going to go into the details of the method at all, but the main point is it quantifies at the level of these splice junctions. So these are the connections between the exons that get spliced together. So leafcutter has been used, for example, by, this is a figure from GTEx, to map these splicing QTLs. So these are associations between genetic variants and splicing variation. GTEx, for example, did this across many different tissues, and we and others have shown that SQTLs are often highly enriched in GWAS. So all these non-coding variants that we find in GWAS for different complex traits, many of them can be explained as acting through effects on expression. We find many of them look to actually be affecting splicing rather than expression. So that raises the question, how much of that splicing that's getting changed is actually functional? One of the ways we tried to look at this is looking at conservation across species. This, for example, is showing the usage of different splice junctions across human tissues. So every row here corresponds to a different splice junction. Every column is a different sample just from human. And you can see these patterns of differential splicing usage across all of these samples. And actually, we see a substantial amount of conservation of these differential splicing differences across tissues when we look across different mammals, for example, here. So these different colors at the bottom are showing you which of the different species these are. And the main point here in terms of the columns is that you're getting clustering by tissue rather than by species, suggesting that there's substantial conservation of this differential splicing variation. But this is evolutionary signal. We think it's valuable, but it's not sort of direct evidence of the function of isoforms. And so some work that we just finally got accepted at Nature Communications after a 2-year review process, which I think is the longest I've ever been through. It's pretty brutal. Was using CRISPR-Cas13. So you probably heard of CRISPR-Cas9, that cuts DNA. Cas13 cuts RNA. And the nice thing for us thinking about splicing variation is that we can target the CRISPR system to specific splice isoforms. So for example, if you have some toy gene here with these 3 different isoforms, you can design CRISPR guides that target the CRISPR system to a specific junction. So for example, all of these guides, these are different little RNA sequences we can design, target the junction between this salmon and this purple exon. And so that when we use these guides, you're only going to get knockdown of this 201 isoform and not 202 or 203, or at least that's the hope. So to test this idea, we did what's called a essentiality screen. So in case you haven't seen one of these before, it's a really powerful general methodology that uses the programmability of CRISPR systems. So the basic idea is that we design a large number of CRISPR guides targeting different genes and are setting potentially different isoforms. And we're going to focus on targeting essential genes. So these are genes where if you knock them down, the cells either die or they at least can't grow. And you can set up your experiment so that the cells that you are treating get, most of the time at least, each cell just gets one CRISPR guide out of this big pool of, in this case, around 50,000 CRISPR guides. Then you let the cells grow for, in this case, we did 3 weeks. And what will happen is the original representation of the guides that you had, you try and aim for them to be even, it's in reality not going to be perfectly even, is going to change over time, because if you have a CRISPR guide which is targeting an essential gene or essential isoform, then that's going to stop those cells that have that guide from growing. And so you're going to see depletion of those guides when you sequence your cells at the later days. So the sort of data that you get out of one of these screens, I'm showing it just for a single guide, so one out of the 50,000 or so that we tested in this experiment. So this is a specific guide targeting a splice junction in an essential gene. And the thing we usually quantify is this log fold change. So this is the fold change in guide count between whichever day I'm showing here, so like between day 7 and versus day 0 when we started the experiment. You can see here that R1 and R2 is we have 2 replicates. So we have these multiple time points, multiple replicates, and this is just for one guide, and we'll have this data for all of the guides. So how could we model this data? Well, we're going to use a Bayesian model, and probably everyone knows what Bayesian statistics is, but just in case, the basic idea with a Bayesian model is that you define, I think I should probably just call this a model, but it's technically a stochastic process, Q, so that's going to be our model, and it has some parameters, theta. And all that we're saying is the data that we observe was sampled from this model Q with parameters theta. And we'd like to know everything we can about the parameters theta given the data that we observed. Bayes' rule tells us how to do that. It says that the posterior distribution, so this is the sort of belief over what the sort of reasonable parameters theta are given the data, is equal to the likelihood, which is the probability of the observed data conditional on some parameters, times a prior that we're going to have to specify, and this kind of annoying evidence term that I will avoid talking about. So how do we represent a model using Pyro? So the traditional, maybe I should just give the context of the traditional way of doing this sort of Bayesian modeling is you get some poor grad student to write down a model, figure out a bunch of math, and then try to write down a way of calculating that posterior distribution. The idea with Pyro is going to be that we just have to write down the model, give Pyro the model and the data, and it'll figure out how to estimate that posterior for us. Okay. This is how we're going to set up the model for Pyro. So it's going to take in as inputs the data. And so weak and observed log fold change here are just the columns of that data frame that I showed you. And there are num_obs observations in the data frame I showed you. That was just 6. And we're going to have to set up a prior on the parameters. We're going to just have one parameter in this model for now, which is called guide score. This is really just going to be the slope of a linear regression linking that log fold change to the time point. And that's, we're going to say, so this is setting up the prior. This says that this guide score parameter is sampled from a normal distribution with location 0, that's just the mean, and scale, that's just the standard deviation of 10. That we could do fancier things than putting 10 as a number, but we'll just pick something for now. Then the actual kind of meat of the model is this is just a linear regression, but it could be any PyTorch code in a more complex model. This could be something like a neural network. So we're going to say the predicted log-fold change is equal to the guide score times the week. So this is our parameter this is a linear regression where this is the coefficient and this is the covariate. So this sort of simplest possible linear model. And then we need to set up the likelihood for the observed data. This plate thing you should think of as just basically being a for loop. So this is a for loop over in our very toy example, 6 observations. And what it says is the sampled or observed log-fold changes are sampled from a normal distribution where the mean is equal to the predicted log-fold change, so the thing we calculated here, the scale, so this is the noise standard deviation. We'll just set that to 0.1 for now, because to keep things simple. And then we can optionally have this observed log-fold change thing can actually be observed, in which case we set this obs input to that observation. And what this does is conditioning, effectively. So this is going to be how Pyro conditions the model on the observed data. And we can also set it to none, and that'll mean that we just sample from this model. We don't normally need to actually return anything from this function with how Pyro works, but just so I can show you that you can actually use this as what's called a generative model, or effectively a simulator, we're also going to return all of the sampled things from the function. Yes, please.
So that how does that connect to it? Like, is it just that's just somewhere like is it the name of the?
Yeah, so it's just the way. So when Pyro actually does its inference later, it's going to know that it's not actually going to be sampling this. It's fixed to this value. So yeah. I think until I show you that how the inference is happening, yeah. But yeah, good question. So this is what's called a generative model, which means we can actually sample from it. One thing we have to do first, which is slight annoyance, is we need to convert. Our data was probably something like a pandas data frame. And so we need to convert it into Pyro works on top of PyTorch, the deep learning framework. So we need to convert our data to PyTorch tensors like this. You could also, at this point, if you wanted to do your computations on the GPU to make it go faster, at this point, you could also specify that you want your data to live on the GPU. I'm not going to show that because it makes it a little more messy. Then if we wanted to sample from the model, which is in the way I think most people here probably think about things is sort of simulating from the model, then we can just call that model function with the observations equal to none. And this is what the data you might end up getting might look like. This looks nothing like the data that I showed you because we haven't trained the model yet. So this will have randomly sampled guide score, i.e. a slope for the regression from that prior normal 0, 10. So that's giving some line like this. And then the actual data points are again sampled from that likelihood distribution. Okay, so how does Pyro actually do its fitting? It uses a thing called stochastic variational inference. I'm going to give almost no details about this, apart from to say the thing we are trying to estimate is this true posterior distribution. The thing is often very messy and complex. In what I've shown you so far, it's actually not. You actually could get it analytically here because it's just linear regression. But a lot of the models we're interested in, this thing is actually pretty complicated and messy. And so what stochastic variational, or SVI, tries to do is find an approximation, Q. Shouldn't have used Q because I used it earlier. Anyway, different Q. So this is an approximation to the posterior that has some nice parametric form. Most of the time, it's going to be a Gaussian, but you could use fancier things as well. And this gets called the variational posterior. It's going to be simpler. And the sense in which SVI tries to make these things too close is a divergence metric called the Kullback-Leibler divergence. It doesn't really matter from the point of view of this talk, the exact mathematical definition of this thing. There are other options that Pyro supports as well, but this is definitely the most widely used. Okay. So a lot of the time, we can get away with using these default variational posteriors that Pyro provides. And the one that we, and I think most people use the most, are these diagonal normal. So this just says that Q distribution is just going to be a multivariate normal with a diagonal covariance matrix. Then part of what we're going to be doing is optimizing that variational posterior, and we do that with it can be any kind of stochastic gradient descent. The most popular is probably Adam, which is also extremely popular for fitting deep learning models. And then we've set up this SVI class, and it needs to know what's the model, what's the posterior, what's the optimizer you're going to use. It also needs to know this loss. This elbow thing is the actual objective function that SVI optimizes. It turns out, I'm not going to go into the details of it at all, but that maximizing this elbow objective is equivalent to minimizing that KL divergence between Q and the unknown posterior. Then we have to do the actual optimization. And so we just iterate like you would if you were fitting a deep learning model. And so we just take these SVI steps. And so the magic you were asking earlier about what happens with observation, the sort of magic that's happening under the hood is that the Pyro machinery is calculating this elbow thing, which includes a likelihood term, and in that likelihood, the observation is set equal to the data. So we can check that this loss function, this loss value is going down over the epochs. It's a bit noisy. That's because the way Pyro evaluates the objective uses Monte Carlo sampling, and so there's noise in the estimation of the elbow. Yeah, please.
What is the parameter that you're trying to infer?
Yeah, so it's a little bit hidden away right now, but what it's actually is so our approximate posterior is a diagonal normal. So in this variational posterior object, there is a mean and a covariance of that normal. And because the SVI has access to the model, what it actually does when you do this call, it runs the model once and it sees what all the parameters are. In this case, it's just that guide score parameter. So it's going to see, okay, there's one parameter called guide score. And so I need just one mean and one variance in this variational posterior to represent like the mean and uncertainty on that parameter.
So the guide score is the parameter?
Well, it's the mean and the variance of the guide score. Because it's Bayesian, it's got to maintain those two things, right? Like it's not just getting an estimate of the guide score; it's getting an estimate of the guide score and its variance. Yeah. So we can extract. There's various. Pyro gives various utilities for extracting the fit from the variational posterior after you've actually fit it. And so one of the easiest ways is this median function. And so if you run that after doing this fitting, you get that it thinks the guide score is like minus 2.2. You can also, to your question, this is how you get the actual mean and standard deviation that's stored in this variational posterior object. And I guess I didn't, yeah, sorry, that's a typo because obviously that should be the same as that. Yeah. Then the other thing you can do is you can sample from the approximate posterior. This is one of the things that I think Pyro could be a bit nicer with how the actual sort of API works, because it uses this predictive function, which is awkward naming, but let's not worry about that. So this is how we would sample 500 samples from the variational posterior that we've fit. We can specify, in this case, this is the only parameter, but you can just specify certain parameters that you want and ignore the other ones, if you so wish. And this is just showing, so each of these lines corresponds to a different sample of the guide score, which corresponds to slightly different fits to the data. We can also simulate from the model once we've fit it. So we can, this function that before we use to get samples of the actual parameters, we can also sample sort of or simulate data from the model once we have that fit. And that's what I'm showing. All these gray points in this little cloud are data points sampled from the fit model. And one issue you can see immediately is the variance is way too small, right? And the reason for that is that we didn't fit the noise of this regression. And that's a pretty weird thing to do if you don't have a good way ahead of time of knowing what the noise variance is going to be. So one of the big advantages of Pyro is it's very easy to change or augment your model. And so, for example, we can add another parameter, which is the standard deviation of the noise. Various different priors you could use for this. Half Cauchy is like recommended by a lot of people, so we'll go with that. And the only thing we need to change in the likelihood is now we're setting, before, if you remember, we set the scale equal to 0.1. Now we're going to set it equal to this parameter. So we didn't have to change the model very much. And we really don't have to change the inference code barely at all. We will do one thing. This is generally a good idea, is we will initialize this model where we're learning the noise to our estimate from the previous simpler model. It's often with these Bayesian models really helpful to give a decent, a sort of sensible initialization to any parameters where you can do that. Now we actually get, so now when we extract the parameters from the model, we also get this parameter for the noise scale, much bigger than 0.1, you'll notice. And now the simulated data does cover all of the data points, but you might actually think we might have overdone it now, right? Like these two data points and these two data points at week 1 and week 3 were actually quite close to our best fit line. It's really just week 2 that looked kind of noisy. And so maybe we're actually like thinking the data is noisier than it really is. This is also showing samples from the model, from the posterior of the guide score. And you can see there's a lot more uncertainty in its estimation now. So those points at week 2 maybe looked a little bit like outliers. Explicitly handling outliers is always a bit annoying, but one thing you can do that's sort of a soft way of handling outliers is to use what's called robust noise models. So before we assumed that the noise was Gaussian distributed, but with Pyro it's very easy to switch out to different distributions. For example, we can use a student t distribution. So this is a family of distributions parameterized by a degrees of freedom, which I'm setting to 3 here. Again, something we could try and learn. And so the student t distribution, if you take this degrees of freedom parameter to infinity, you get the Gaussian distribution back. But if you have smaller values of these degrees of freedom, you get a distribution that has much heavier tails. And so then the model is less surprised by things that look like outliers. So inference code wouldn't have to change. We can refit this. And this is showing what the data looks like under that model fit with the robust noise. And this is the estimates of the guide score, the mean and standard error effectively. So under the Gaussian noise model, you can see we had quite a big variance on this estimate of the guide score. And that actually goes down when we use the robust noise model. So this was all fitting data just for one guide, but we have 50,000 guides. Good. And we are also asking a lot of 6 data points, right? I'm trying to fit a noise variance and a mean, and maybe I'd like to have fit that degrees of freedom parameter as well. And so ideally, we would like to fit data for all of the guides, and maybe we can also use the fact that we have all this data to give us improved estimates for individual guides as well. And so this idea gets some different names. I think of it as sort of sharing statistical strength between different parts of the data. It also has a technical sort of name for this approach, which is called empirical Bayes. So we can think of a few basic strategies we could take to handling all of our different guides. So the simplest one would be to have just fit a separate model for every single one of those 50,000 guides. On the other extreme, I could say, I think the amount of noise that I have in this experiment, it was one big pooled experiment. So maybe the noise should be the same across all of the guides. So I could just fix the noise scale to be the same. But maybe the ideal would be to find somewhere in the middle, some optimal point where I share some information. I say probably the noise variances are sort of similar across guides, but not exactly the same. So empirical Bayes lets you do this. And the way it does it is to say, I'm going to learn a prior distribution on the noise scale so that prior is shared across all of the guides. And then for each individual guide, there is a different noise scale, but they're encouraged to be similar through this prior distribution. If I learn that that prior distribution is very narrow, that's sort of pushing me more towards this end of saying all of the noise variances are very similar. If I learn that that prior distribution is very wide, that's saying all of these noise variances are very different across the guides. So I have to set up the data across all of the different guides. I won't go through this in detail. I guess I will just say, so one thing we're going to get is we're going to now have a big data frame that includes all of the guides. And so for each row, there's going to be an index, a guide index, which says which guide did this data point come from. I think that's the key thing here. And so then we can have a model that goes across all of the guides. And these are the parameters of that prior on the noise scales that we're going to learn. I think it's actually probably helpful if I show this first. So remember, this plate thing is like a for loop. So this is a for loop over all of the guides. And it samples a guide score. So we've already seen that that's saying exactly the same as we had for the single guide model. But because it's within this for loop, there's a guide score for every guide. And then there is now a noise scale for every guide. And that noise scale is sampled. It needs to be positive. So instead of sampling it from a normal distribution, we sample it from a log normal distribution. A log normal distribution is just what you get if you take a normal distributed variable and then you exponentiate it. So now it has to be positive. And the mean and standard deviation of that normal distribution is given by these two parameters, these two new parameters that I introduced that are shared across all of the guides. The regression model part is basically the same, except for each guide that I'm looking at, I just need to, for every data point, every row of my data frame that I'm looking at, I just need to pull out the correct guide score for the row that I'm looking at. And then the likelihood is very similar to what we had before. But again, now I need to index, just like I did here, I need to index the correct noise standard deviation for the specific guide that I'm looking at for that row. So this still fits actually pretty fast, even on CPU. It took about 40 seconds to fit all of this data. It would be faster if you moved it to GPU as well. And what I'm showing here, so this is the distribution of the guide scores across all of the 50,000 guide RNAs. You can see there's a bunch down here with quite negative values. So these are going to be the guides that actually worked successfully. They successfully knocked down the essential gene that they were targeting. But you'll notice there's a ton of stuff around 0. So these are all the guides that actually didn't work. And we can look at the learned prior distribution on the noise scale. So this red line is showing that log normal distribution after it's been fit on the data. So the mean ended up being minus 0.5 and the standard deviation 0.3. And the blue here is the distribution of the actual estimated noise scales across all the guide RNAs. You can see it looks like there's a bit of a discrepancy. That's because this is just the blue is showing the posterior mean for every noise scale. And then there's going to be some uncertainty in that, and that's what the red is taking into account. And I am roughly out of time, so I will just say that the actual model we used in the end is very close actually to this. There's a few more extensions. You can look at it in the finally published or accepted paper. And this point that a lot of CRISPR guides don't actually work is part of why we developed this model with Neville Sanjana's lab that's a deep learning model that predicts based on sequence which guides will work. So if you're doing CRISPR screens, you really need to actually select ahead of time guides which are going to work well. I will leave this up and end there, since this is maybe good like questions and discussion type material. Anyway, yeah, we actually get all of these to come up, and I will stop there roughly on time. Thanks.